Welcome to another voyage. This is a quiet place, where the noise of the day fades and the deeper rhythms of life begin to rise. Here we trade questions for wonder and facts become poetry in motion. You're not alone, you're adrift in the great unfolding of the living world. Tonight we journey inward, not into the cosmos above, but into the cosmos within. A hidden world, microscopic and miraculous, where every heartbeat, every breath, every thought is powered by molecular machines, too small to see, yet too vital to forget. We'll wander the winding highways of the cell, watching tiny couriers carry messages and cargo. We'll listen to the ribosomes' quiet song, as it weaves chains of amino acids into the fabric of life. We'll explore the glowing mitochondria, where fire is made from breath, and energy becomes motion, memory, healing, and thought. This is a story of threads and folds, of shape and function, of ancient chemistry, still unfolding in every cell of your body. And if you'd like to drift deeper into sleep, softly, without interruption, it's best to watch this video as part of the full Midnight Voyager playlist. That way the stories will flow one into the next, like gentle tides of thought, carrying you through dreamlike journeys, while the world quietly fades. Before we begin this inward voyage, I'd love to hear from you. What time is it where you are? Are you resting in the hush of midnight, stirring in the stillness of 3 a.m., or waking early beneath the quiet songs of birds? Let us know in the comments. You're among fellow travelers here, a community of minds who drift through science, not just to learn, but to feel something deeper. Now settle in. Let your body rest, your eyes grow heavy, and your thoughts soften into stillness. Together, we begin tonight's midnight voyage into the cell, into the story of life. Close your eyes for a moment and imagine floating. There is no weight here, no boundary, no wind, only stillness. Soft, quiet, like the breath before a dream begins. Around you, the world holds its shape, just as it always has. And yet, beneath that surface, something begins to shift. The space no longer stretches outward. It draws inward, slowly, gently, as if the entire universe is folding into itself. And you are falling with it, not with speed, not with fear, but with a widening sense of wonder. You are shrinking, not in body, but in scale, shrinking beyond sight, into the unseen. The freckle on your hand rises like a soft hill. A single strand of hair becomes a waving beam of light. The grooves of your fingerprints stretch like distant ridges, then fade. Down you go, through the skin surface, beneath layers of keratin and collagen, past rivers of blood cells rushing like crimson boats along branching canals. You slip between fibers, glide through membranes, float along currents of salt and sugar. Swirling through the interstitial sea, structures begin to loom. Once invisible, now immense, cells rise before you, not as drawings, but as living, soft-walled worlds. Their membranes ripple with life. Inside, tiny bodies spin and swim, and the boundaries of self begin to blur. You drift closer. A single cell, once smaller than a speck, now fills your vision like a cathedral. Its outer wall pulses gently. Its contents shimmer with purpose. Molecules dart, fold, and collide. Here nothing is still, and yet everything is precise. You are not dreaming, you are arriving. This is a journey through the hidden machinery of life, a descent, not into mystery, but into intimacy. Here, in the smallest parts of you, lie the oldest instructions. Every breath you take is born from movements too small to see. You've reached the threshold of the invisible. And just beyond this veil, the cell awaits. To walk among its structures, we must first leave behind the scale of the everyday. You are used to the visible, mountains, trees, the width of a river, the blink of an eye, even the smallest things you know. A grain of sand, a droplet of mist, the width of a hair, still belong to a world shaped by human sense. But here in the realm of the cell, we cross into a world ruled by scale, where size means something entirely different. A single human hair is about 80,000 nanometers wide. It's just a number. Big on paper, invisible in life. Now imagine this. Inside one human cell are ribosomes, tiny machines that build proteins, each one only 20 nanometers across, thousands of them in every cell, so small that even a virus would feel crowded beside them. Your DNA, the great script of your being, is only 2 nanometers wide, so narrow it could rest inside a groove. Of a groove, and yet, that same strand of DNA, if stretched out from just one cell, would be nearly 2 meters long. Think of that. 2 meters of instruction, folded into a space no eye could ever see. Mitochondria, the cell's engines, are about 1,000 nanometers wide. Microtubules, its internal roads, span just 25. Water molecules are less than 1. But in this world, size does not limit meaning. Here, shape is destiny. A misfolded protein, just a few nanometers wide, can change the life of a cell. An ion drifting inward, calcium, potassium, sodium, can signal a heartbeat, a thought, a memory. Nothing is wasted, no movement is meaningless. Even the smallest flicker can ripple outward, touching sensation, affecting perception. The cell is not a bag of parts, it is a vast humming system. Its logic lives in the folds of proteins, the tides of ions, the softness of membranes. To truly see it, we must let go of size and begin to feel significance. You may have seen it in a textbook, a circle for the nucleus, some beans for mitochondria, a few dots labelled ribosomes, so clean, so simple, but the truth is far more alive. Step inside a real cell, and nothing is still. There is no emptiness and no open space. The interior is crowded, brimming with life. Molecules drift and bump. Proteins fold and unfold. Enzymes search with quiet urgency. Vesicles bud and travel. Tiny ships crossing a crowded sea. There are over 20,000 kinds of proteins in a human cell, each one folded just so, each with a task. Some act like scissors, cutting molecules in precise places. Others are lifters, movers, builders. Some carry messages from the outer membrane to the inner core, triggering change in moments. The cytoskeleton, the cell's frame, stretches like a web beneath it all, not rigid but alive. It builds, it breaks, it reforms, responding to the cell's needs in real time. Nothing is random. The cell listens, adapts, repairs, recycles, and when it must, divides. It creates a copy of itself, whole and new. All of this happening in a space smaller than a grain of dust. And yet its complexity rivals our most advanced machines, perhaps even surpasses them. To call it simple is to call a city a scribble on a page. Now we hover at the edge, the final veil, not a wall, but a living membrane, a soft, shifting surface of phospholipids. It is the cell's gatekeeper, its shape, its guard, its guide. It does not sleep. Proteins embedded within act as sentinels, channels, receptors, carriers. Ions drift through in careful patterns. Messages land like raindrops. The cell listens and responds. Behind that gate lies the cytoplasm, the sea within, busy, moving, alive. Organelles float an anchor. Energy is made, instructions are read. Molecules are built with purpose. It never stops. The cell reshapes itself moment by moment. A city reimagined with every breath. As you pause here, at the edge of this living world, know this isn't metaphor. This is you. Every muscle, every nerve, every drop of blood is made of cells just like this one. You are not a single being, but a constellation, a gathering of life working together to make you. You are built not from stone or steel, but from rhythm and code, from motion and intention. You are biology, made real. And now, we are ready to cross the threshold, to step inside, to explore the city within. The journey has only just begun. Now that we've passed through the threshold, let us step fully inside. What lies ahead is not a blur of chemicals, and not an empty pouch of fluid. It is a place of structure and rhythm, of motion and meaning. You are not in a void, you're in a city. A cell is like a city, though no rooftop or stone wall could match its grace. Its architecture is soft, flexible, alive. The streets are lined with filaments of protein. The buildings, not brick but folded membranes, sculpted compartments, floating spheres of function. The city's edge, the plasma membrane, is more than a border. It's a conversation, a living dialogue between inside and out. It ripples softly with each chemical change, studded with proteins, acting as gates, locks, and listening posts. It's selective, curious, always watching. It lets in what's needed, nutrients, ions, messages, and bars what would do harm. It keeps the cell whole but never closed. Inside these walls lies the foundation, a molecular framework called the cytoskeleton. These are not bones in the usual sense. They're flexible, restless, always rebuilding. Long strands of actin stretch beneath the surface, like cables beneath a tent. Microtubules radiate outward like spokes from a hub. Intermediate filaments hold everything steady. But they do more than support. They allow the cell to reach, to reshape, to divide. If the membrane is the border, the cytoskeleton is the infrastructure. The beams, bridges, and highways that make life possible. And yet, unlike steel or stone, this structure is alive. It assembles and disassembles in real time, shifting as the cell moves, adapting with each moment. Built into its form is wisdom, not thought, but design, a living structure perfected by evolution. This is the frame and the shape upon which the cell will build its life. And deeper still, past these walls and threads, lie the districts, each with their own role. Step further in, and you'll see it clearly. This is not one open space, but a landscape divided by purpose. Each region, each fold, every floating body has its place. It is a city of neighbourhoods, each busy with its own rhythm, all connected by invisible lines of communication. At the centre is the nucleus, the archive, the deep library, the district of identity, spherical and quiet, wrapped in its own double membrane, not isolated, only protected, a sanctuary for DNA. Inside, chromatin coils and loops, storing the full genetic code. But this library is no museum, it's active. Genes are read here, instructions copied, blueprints drawn for every protein in the body. Nearby, the terrain changes. Folded membranes stretch outward. The endoplasmic reticulum, or ER, the rough ER, studded with ribosomes, is a workshop where proteins are built from RNA's instructions. The smooth ER is calmer. It makes lipids, processes toxins. Both together form the city's industrial zone, assembling and refining the cell's raw materials. Just beyond, you'll find the Golgi apparatus, a stack of flattened sacks shimmering like paper. This is the post office. Proteins and lipids arrive here in vesicles. They're tagged, modified, sorted, each one sent to its correct destination. Some to the membrane, some to storage, some to the world beyond. Nothing is sent without a label, nothing without a reason. Scattered throughout the cytoplasm are lysosomes and peroxisomes, the city's recyclers, its detox chambers. Lysosomes break down waste. They dismantle the old and the broken. Peroxisomes neutralize harmful chemicals. Together, they keep the city clean. Quiet custodians sweeping the floor, each organelle a neighbourhood, each district alive with purpose. And though none of them speak, they understand one another. Signals pass like breath. Their rhythm is the heartbeat of the cell. Because every city must move, and here nothing stands still. Inside the cell, motion is not a luxury. It is life. Molecules, vesicles, organelles. They do not drift aimlessly. They're guided, directed, purposeful. Along the cell's highways, the microtubules. Cargo is carried by motor proteins. These are kinesin and dynein, walking step by step along the protein rails. They move like tiny figures, each with 2 feet, hauling vesicles as if pushing little carts. Each vesicle holds cargo, a protein ready to ship, waste bound for recycling, or a message, about to be released. These motors do not think, they don't choose, but because of them, the city works. Efficient, responsive, alive. And just as the cell moves things through space, it moves information. Signals arrive from the outside. Light, heat, hormones, whispers of the world. Receptors in the membrane listen. When a signal is heard, a cascade begins. One molecule activates the next, until a gene is turned on, or a gate is opened, or a message is delivered. There is no brain in the cell, no overseer, yet it reacts with intelligence, because the message always flows from vesicle to signal, from edge to core. The cell is always moving, always listening, always speaking. The cell never sleeps, even as you lie still, eyes closed, drifting into dreams. The cell continues, it builds, it repairs, it speaks. Proteins are born, folded, assembled. Membranes reshape, vesicles deliver, ions flow. The nucleus receives word. Genes are turned on, turned off, tuned like an orchestra. Damaged proteins are flagged. Misfolded chains, escorted to the proteasome, taken apart gently, rebuilt with care. And always the cell listens, not with ears, but with balance, not with words, but with chemistry. It adapts, it heals, it keeps its place in your greater body, deep, in blood, in brain, in skin and bone, even as you sleep. It goes on. This city is not made of steel, it is built from fluid, from folded chains, from membranes and rhythm and code, and you are not apart from it. You are this city. Its breath is your breath, its rhythm, your rhythm. As we leave now, this cell, this world within, carry its wonder forward. Know that within you, life continues, perfectly, patiently, beautifully unseen. At the heart of every cell lies a structure unlike any other, quiet, round and profound, the nucleus. From the outside it appears simple, like a pearl suspended in mist. But within its boundaries lies something vast, a complete record of what the cell is, and what it may become. The nucleus is not merely a centre, it is a sanctuary, wrapped in a double membrane, dotted with pores like tiny guarded gates. It keeps its contents protected, yet accessible. And inside, drifting in a soft, dense fog, coils the DNA, the molecule of life. The spiral you may have seen, elegant and simple in drawing, is in truth far longer and far more intricate than one might imagine. Stretched end to end, the DNA inside one nucleus would reach nearly 2 meters. And yet it is folded, looped and wrapped so finely that it fits into a space invisible to the human eye. But it is not just folded, it is organized. Genes are stored in regions. Loops of DNA are tethered to internal scaffolds, not randomly, but with purpose. Some regions lie silent, tucked away like archived memories. Others are open, active, fluttering with molecular activity. These are the pages being read, because the nucleus is not just a vault. It is a library, and in this library the cell reads instructions. It copies only what it needs, when it needs it, using enzymes like scribes, to transcribe select stretches of DNA into RNA, molecules that carry the message outward, into the body of the cell. Not every gene is read in every cell. A muscle cell reads one set, a neuron another, a liver cell something else entirely. Yet all carry the same book. The difference lies in which pages are opened. This selective reading is what gives the body form and function. Eyes and skin, bone and blood, all shaped from the same master text, but read in different ways. The code itself never speaks, but the cell knows how to listen. The DNA within the nucleus does not speak in sentences, but it tells stories all the same. Its alphabet is built from just 4 letters, A, T, C, and G, short for adenine, thymine, cytosine, and guanine. They pair with one another, like matched lines of poetry. Are always binds with T, C with G. Across the double helix, they form rungs on a spiraling ladder, and in their sequence they encode life itself. A gene is not a single idea, but a stretch of code, some long, some short, each one like a paragraph within a chapter. Some genes direct the building of proteins, others regulate when those proteins are made. Some remain unread for years, waiting in silence until the cell encounters a stress, a signal, a time of need. And between these genes, spaces. Once thought to be meaningless, these non-coding regions are now understood to be full of purpose. Like the margins in a notebook, they hold notes, boundaries, timing cues. They determine which pages of the genome are folded open and which remain sealed. But the code alone is not enough. It must be read. When the cell requires a specific instruction, it initiates transcription. The DNA at that location unwinds slightly, and an enzyme called RNA polymerase attaches itself to the strand. Like a scribe copying from a scroll, it builds a matching strand of messenger RNA, or mRNA, letter by letter, word by word. This RNA is not permanent. It is a working message, a disposable copy. When complete, it exits the nucleus through one of its guarded pores, carrying the instruction into the cytoplasm. There it will be read again, this time by ribosomes, and translated into a protein. From one library, the message is carried. From one message, a structure will be built. This is the quiet miracle at the heart of every cell, a language that does not speak aloud, yet is always heard. The genome, vast as it is not left unguarded. It is monitored, maintained, and meticulously repaired, not by one watchful mind, but by many. Proteins patrol its coils, enzymes check for errors, signals flash when damage is found. Because DNA, though powerful, is delicate. It can be broken by radiation, mutated by chemicals, copied imperfectly in haste. A single mistake left unchecked might lead a cell astray, into malfunction, into silence, or into disease. And so the cell invests in protection. There are proofreaders, molecular sentinels, that double-check every letter copied during replication. If a mismatch is found, they pause, remove the error, and insert the correct letter with steady hands. There are repair enzymes, standing ready to mend broken strands, to excise damaged sections, and rebuild them using the untouched half as a guide. And there are histones, the proteins around which DNA is wrapped. Not just structural supports, but regulators too. They decide which sections of the genome are accessible, and which are not. These layers of control go deeper still. Some genes are silenced not by mutation, but by epigenetic marks, chemical tags placed upon the DNA or its histones, telling the cell when a gene should be active, and when it should remain quiet. These marks shift across a lifetime, influenced by environment, experience, even memory. The genome is constant, but its expression is a dance. And when all else fails, when damage is too deep or danger too great, the cell may choose to let go, to initiate a calm and silent end, so the whole may be protected. This process is called apoptosis, programmed cell death, and it is not violence, but wisdom. Within this nucleus, then, lies not just the script for life, but the means to protect it, to pause it, to preserve it, to pass it on. And all of it happens in silence, inside every cell, inside every one of us, inside the quiet shelter of the nucleus. A decision is made, not by a voice or a command, but by molecular need, a signal received, a cue from without or within. A gene nestled deep within the coils of DNA begins to stir. The cell has need of something, a new protein and a structural repair, an enzyme for the work ahead. And so the scribes awaken. DNA is sacred, irreplaceable, too important to be used directly. Instead, the cell sends its copyists. One of them, RNA polymerase, approaches the gene with care. The DNA at that site begins to unwind, just slightly, a small opening in the great spiral staircase, enough for the scribe to begin its work. It attaches itself to one strand of DNA, the template strand, and begins to read, one letter at a time. It moves forward, assembling a matching strand of RNA. A with U, C with G, G with C, T with A. This new strand is messenger RNA, or mRNA, a transcript, a mirror of the gene's message, built from single-stranded molecules that will soon carry instructions beyond the nuclear wall. The process is deliberate. The polymerase moves steadily, not rushing. Behind it, the DNA coils closed again, and before it, the new message grows. A thread of meaning, spun in silence. This transcription is not random. Only the needed genes are transcribed, only the right messages, at the right time, in the right amount. And when the polymerase reaches the end of the gene, it stops and releases the finished message into the nucleus, where the next part of its journey will begin. The message is written, but it is not yet ready. Before the mRNA can leave the nucleus, it must be prepared, polished, protected, and refined. The nucleus is not simply a vault for DNA. It is also an editing room, a place where every message is shaped for its task. First, the cap is placed on one end, a special structure that marks the message as complete and trustworthy. It's more than a decoration. It tells the cell, this belongs here. It guides the mRNA to the ribosomes, ensuring it is recognized and read correctly. Next, the long tail of adenine bases, a poly-A tail, is added to the other end. This tail acts like a fuse, slowly shortening with time. The longer the tail, the longer the message will last. It is the cell's way of timing the life of each instruction, giving it time to be read. But not forever, because in the cell, nothing is permanent. Only the rhythm remains. Then comes the most delicate step, splicing. You see, the message as first transcribed contains more than just instructions. It holds interruptions, sections that do not code for protein. These are called introns, and they must be removed. Between them lie the exons, the true coding sequences, the parts the ribosome will read. A molecular machine, known as the spliceosome, finds these introns, loops them out, and cuts them away. The remaining exons are stitched together, forming a clean, continuous script. This editing is precise, and sometimes it is creative. For in some cases, the cell splices the same gene in different ways, producing more than one message from a single piece of code, a phenomenon called alternative splicing. From one gene, many proteins can emerge, depending on how the message is cut and rearranged. It is a kind of molecular improvisation, a flexibility built into the very language of life. And now, with its cap, its tail, and its edited form, the message is complete. It floats gently through the nuclear fluid toward one of the many pores that dot the membrane, and then, like a letter sent into the world, it passes through, into the cytoplasm, into the body of the cell, into motion. The message slips through the pore like a breath escaping a chamber, from the nucleus into the cytoplasm, from silence into the hum of the cellular city. The cytoplasm is crowded. It is not empty space, but a sea thick with motion. Vesicles gliding, proteins folding, signals flashing in ripples too fast to see. And into this sea, the messenger RNA now floats, a delicate strand of instruction, coated in protective proteins, guided by hidden cues. It is not lost. It knows where to go. Or rather, the cell knows. Scattered throughout this inner ocean are ribosomes, tiny machines, ancient and tireless, waiting for messages to read. Some are free floating, drifting like buoys in the cytoplasmic tide. Others are docked to the rough endoplasmic reticulum, ready to synthesize proteins bound for membranes or export. But all of them serve the same purpose, to read the message and translate it into substance. As the mRNA approaches, the ribosome assembles, two halves joining like clasped hands. One half cradles the message, the other waits to begin. And then, without ceremony, the first codon is read. 3 letters. That's all it takes to start. The ribosome matches those 3 letters, that codon, with a molecule of tRNA carrying the right amino acid. A match is made, a bond is formed. And just like that, the first building block of a new protein is set in place. The message is no longer just a thread of code. It is becoming something real. Letter by letter, codon by codon, the language of life is being translated into form. The ribosome reads 3 letters at a time. Each triplet a codon, each codon a signal for one of 20 amino acids. The small versatile molecules that make up every protein you've ever known. With each new codon read, a new tRNA arrives. Its anticodon perfectly matched, its amino acid offered forward like a gift. The ribosome binds it in place, then forms a peptide bond, linking this new amino acid to the growing chain. A polypeptide, slowly lengthening, folding even as it grows. This chain is more than a string. It is a future structure, a potential enzyme, a channel, a receptor, a scaffold. It might bind iron, it might carry oxygen, it might ferry signals between neurons. But for now, it is becoming. The ribosome moves along the message, step by step, rhythmically, patiently. It reads until it encounters a stop codon. A signal that the message is complete. Then, the ribosome lets go. The mRNA may be read again, or degraded, its purpose fulfilled. The newly formed protein floats free into the cytoplasm. Its destiny just beginning. Some proteins will stay close to where they were made. Others will be folded and packaged, shuttled to the Golgi, tagged for export, sent to membranes, to organelles, even to the spaces between cells. Some will fold into shapes so precise they will trigger the contraction of a muscle. Others will catch photons of light, allowing you to see, but they all began the same way, from a stretch of DNA, to a strand of RNA, to a chain of amino acids. Spoken into being by a ribosome that never sleeps, this is how information becomes form, how language becomes life, how the cell turns silence into substance. Of all the machines inside the cell, the pumps, the motors, the messengers, none hold quite the quiet elegance of the ribosome. It is not large, it is not fast, but it is tireless and profoundly essential. It is the ribosome that sings the proteins into being, not with voice, not with thought, but with motion, with precision, with rhythm. It is ancient, born in a world before complex cells, before nuclei, before mitochondria, before membranes as we know them. The ribosome has been with life since nearly the beginning, a relic of a time when RNA alone may have ruled, and proteins were just beginning to shape the world. Even now, every living cell, in every tree, every animal, every single-celled organism, carries ribosomes. They are the great unifiers, the common thread, the universal machine. They are made of 2 subunits, one large, one small, both composed of ribosomal RNA and protein, folded intricately, almost like origami in motion. And when the message, the messenger, RNA, arrives, these 2 halves join together around it, forming a tunnel. A channel through which information flows and structure is born. They do not hesitate, they do not falter. They begin to read. Each codon is an instruction. Each instruction summons a trana, carrying with it an amino acid, the next note in the song, and the ribosome binds them together, link by link, like a composer writing melody on a staff of silence. But this melody is not sound. It is shape, function, future. The ribosome does not know what it builds. It does not need to. It simply plays its part. The quiet composer within. Within the ribosome, the work continues, calm, deliberate, uninterrupted. One by one, the amino acids are linked into a growing chain. Each one is chosen with care, matched to the message, placed in the precise position, like beads sliding onto a thread that knows exactly what it's becoming. This chain is not yet a protein, and it has no shape, no purpose. It is raw material, potential. But even now, within its length, the instructions for folding are encoded. Each amino acid carries a personality. Typed, some seek water, others flee from it. Some are charged, others neutral. These differences are not flaws, they're instructions. As the chain grows, it begins to curl and bend. Loops and folds emerge, soft spirals called alpha helices, flat planes called beta sheets. These are the first signs of structure, guided not by hands but by physics, by charge, by chemistry. Often the cell assigns chaperones, proteins whose sole task is to guide the folding process, to shield the growing chain from interference, to ensure it folds correctly and not too soon. Without this care, the chain could collapse incorrectly, as they twist into shapes that cannot function, or worse, begin to clump, forming sticky tangles that gum up the machinery of the cell. But when all goes well, when the sequence and the environment are just right, the chain folds itself into a final, three-dimensional form, as precise as a key, as unique as a snowflake, as necessary as breath. It is no longer a chain, it is a protein, and it is ready. And it all began with a ribosome and a single line of code. Now the chain has folded, the protein is whole, but even now the story is not finished. A structure alone is not enough. It must be placed, delivered, positioned within the vast and crowded cell where it is needed most. Some proteins stay close to home, drifting freely in the cytoplasm, where they act as enzymes, messengers, builders. Others are born at ribosomes bound to the rough endoplasmic reticulum, their destination already set. There they are threaded directly into the ER's folded membranes, a quiet corrugated landscape where finishing touches are applied, chemical tags are added, sugars are attached, folding is refined, misfolded proteins are recognized, retained, even discarded, because only the perfect structures can move forward. From the ER, the protein is packaged into a vesicle, a small sealed bubble, and sent onward to the Golgi apparatus, that elegant stack of membranes folded like a set of ribbons in space. The Golgi does more than receive. It decides, it modifies, sorts, and ships each protein with precision. One protein may be bound for the membrane, to become a channel, a receptor, a gate. Another may be headed outside the cell entirely. A hormone, perhaps, or an antibody, waiting to be released. A third might be delivered to the lysosome, to help break down waste. Each destination is marked with molecular codes, readable only by the cell itself, a language of sugars and folds, of position and shape. And so, with its journey nearly complete, the protein leaves the Golgi, ready to take its place in the living machine. It began as a message, a fleeting thread of mRNA. Now it is a tool, a worker, a structure. It is a finished song, set loose into the world. Now the protein has found its place. Its shape is correct, its journey complete. And with that, it begins to work. Some will catalyze reactions, enzymes that lower the barriers to change. They take the impossible and make it easy. They break molecules apart, or stitch them together with speed and elegance, over and over again, without tiring. Others will serve as channels, tunnels through the membrane, opening and closing to let ions in or out. They help the cell maintain its balance, its charge, its rhythm. Some proteins form scaffolds, cables and beams of cytoskeletal structure, giving the cell its shape, allowing it to stretch, contract, or crawl. Still others will be sent beyond the cell itself, floating in blood, carrying oxygen, or acting as signals between neurons, whispers between cells in the vast and silent body. Some are structural, keratin in your hair, collagen in your skin. They give form to the flesh. Some are defenders, antibodies patrolling your bloodstream, ready to bind to invaders, marking them for destruction. And some are messengers, hormones drifting through the body, like distant songs, each one carrying a message, each one seeking the right listener. All of them, everyone, born from the same process. A ribosome reading a message, a chain of amino acids folding, a destination, a function, a life. This is the machinery of biology, not made of metal, but of softness, of motion, of purpose written in molecules. And it never ends. As you rest now, dozens of proteins are being born inside you, right now, this moment, in every cell, in every organ, in quiet harmony. They will build, defend, listen, move. They will hold you together. They will keep you alive, And all of it began with the ribosome song, a melody without sound but full of meaning. Imagine a single thread, no wider than a whisper, uncoiling in darkness, born not of cloth or silk but of atoms, linked end to end in perfect order. This is a protein at its beginning, a chain of amino acids freshly forged by the ribosome, still linear, still soft, still undefined. But this is not its final form. It is only the raw material of what it is meant to become. Proteins are not useful because of their length or their ingredients, but because of their shape. Shape is everything. A protein's fold determines whether it binds or breaks, whether it carries or catalyzes, whether it helps life bloom or falter. And so, even as the chain emerges, it begins to twist. Some sections coil into spirals, alpha helices, like ribbons caught in a breeze. Others flatten into beta sheets. Folds aligned in calm, pleated precision. These early shapes are not imposed from without. They are written into the chain itself, a hidden logic in the sequence where hydrophobic regions tuck themselves inward and polar parts reach toward water. The forces at play are quiet, but relentless. Electrostatic attractions, hydrogen bonds, the push and pull of molecular friction. And yet, the result is grace. From a random coil emerges structure. From structure, function. Like origami guided only by the nature of the paper, the protein folds itself into a three-dimensional sculpture. Delicate, but exact, a lock ready to receive its key, a hand extended into the world. And in this folding, the cell transforms a simple chain into something capable of thought, motion, memory, or healing. Folding, though elegant, is not always flawless. Even the most finely tuned sequences can falter, especially in the crowded, shifting environment of the cell. Here, amidst a sea of molecules, a freshly formed protein is vulnerable, prone to misfolding, to sticking, to collapse. But the cell is not unprepared. It assigns guardians to this fragile task. Silent watchers known as chaperone proteins. They do not change the message, they do not write the code, but they guide the fold. Some chaperones hover nearby, offering protection, preventing premature interactions. Others form enclosed chambers. Molecular sanctuaries where a single protein can fold in peace, uninterrupted, unseen. These chambers, called chaperonins, are among the most remarkable structures in the cell. Cylindrical, capped, precise. They take in the newborn protein, shield it from the world, and give it space to find its correct form. There is no forcefulness, no reshaping by hand, only gentle constraint. And time. When folding is successful, the protein is released, shaped, tested, and ready to serve. But not all folds succeed. Some chains twist into the wrong shapes. Some stick together in dangerous clumps. These misfolded forms can be harmful, interrupting processes, disrupting membranes, even leading to disease. And so the cell must choose. It tries again, or it begins to dismantle, sending the protein to the proteasome, the molecular recycler, where the chain is broken down into its parts, ready to be reused in a future fold. This quiet system of trial, error, and repair is not infallible, but it is persistent. And within it lies one of the deepest truths of life. That function is born not only from code, but from care, from the way a chain is folded, from the keepers who watch, from the balance between order and entropy. This is the art of biological origami. Even in the most elegant systems, failure finds a way. Protein folding is not merely a task, it is a risk. And when that risk is unmet, the results can ripple across the cell, across tissues, even across time. A misfolded protein is more than a mistake. It is a danger, a shape the cell cannot use but cannot always ignore. In the best case scenario, the faulty chain is caught early, marked with molecular tags, and escorted quietly to the proteasome, where it is unwound and dismantled, its amino acids set free to be used again. But sometimes the fold goes unnoticed, or worse, it persists. and begins to attract others. Misfolded proteins have a tendency to stick to themselves, to others, forming aggregates, clumps, plaques. These are the scars of failure, seen in diseases like Alzheimer's, Parkinson's, and Huntington's. Here, the proteins do not simply cease to function. They begin to interfere. They crowd, they disrupt, they linger, and still the cell tries to correct them. Chaperones return, attempting to rescue the shape. Autophagosomes may form, encasing the aggregates and sending them to the lysosome, hoping to cleanse the city before damage spreads. But the effort is not always enough. Some misfolded proteins resist destruction, like a riddle with no solution. And yet, even in failure there is insight. The study of misfolded proteins teaches us not only about disease, but about the fragility of life's architecture, about how tightly woven the balance is between function and dysfunction. Folding is not guaranteed, but it is guarded, and when it goes wrong, it reminds us that perfection is not the absence of error, but the presence of systems that try again and again to get it right. What gives something meaning? In the world of proteins, it is not what they say, but how they fold. Their story is written not in paragraphs or pages, but in loops, spirals, pockets, and planes. A folded protein can cradle a metal ion. It can break a molecule in two. It can carry oxygen from lung to limb. It can open a channel in the neuron's wall and let thought pass through. It can bind to DNA, to light, to sugar, to scent. It can recognize self or signal danger, all from a single chain, curled just so. This is the miracle of biological origami. That sequence becomes shape, and shape becomes function, and function becomes sensation, thought, memory, movement, healing. You are held together by folds. You think in folds. You remember in folds. The enzymes in your saliva, the antibodies in your blood, the pigments in your eyes, each of them born from the delicate precision of folding. And yet it happens without noise, without light, without witness, inside you now, even now. Tens of thousands of folds are forming, chains fresh from the ribosome, coiling and curving, each with a purpose already hidden in its form. No two folds are exactly alike. Even the same protein may take subtly different shapes. There is room for variation, for motion, for life. And from this quiet craft, the body is built, not from bricks or beams, but from threads. Folded in darkness, lit only by meaning. All life requires energy, to move, to divide, to heal, to remember, even to sleep. And yet the cell has no batteries, no wires, no generators in the way we understand them. Instead, energy flows through the body like a secret current, carried in molecules, unlocked by enzymes, sparked into motion by ancient chemistry. It begins with food, but more precisely, with the glucose held within it, a simple sugar made by plants from sunlight, stored in fruit, in grain, in root and seed. When you eat, glucose enters your bloodstream, then your cells, where it becomes the starting point for a quiet chain of reactions. This process is called glycolysis, and it takes place in the cytoplasm, the open interior of the cell, where proteins float and organelles drift. Step by step, the glucose molecule is broken down, split into two smaller units called pyruvate. In the process, the cell captures some energy in the form of a molecule known as ATP, adenosine triphosphate, the cell's temporary flame. But this is only the beginning. The yield is small, just enough to prime the system, to send a signal to the mitochondria, waiting deeper within the cell, that more energy is needed. Glycolysis is like striking a match, a brief flicker. That prepares the way for the fire to come. And so the pyruvate drifts inward, toward the powerhouse, toward the ancient organelle that lights every living cell. With the flame of its breath, the pyruvate born from glucose drifts through the cytoplasm, drawn inward, toward an organelle that is not just another structure, but a relic, an ember from a time before multicellular life, the mitochondrion. It is more than a machine. It is a symbiont, once an independent bacterium, now woven into the fabric of every animal, every plant, every human cell. A partnership written into our lineage, so ancient that its memory lives on in the spiral of its own DNA, surrounded by two membranes, an outer one smooth and simple, and an inner one folded into deep curves called cristae. The mitochondrion is a world within a world. The pyruvate crosses the first membrane easily, but the inner membrane is selective. It holds tight control over what may enter. Here the pyruvate is transformed again. Stripped of a carbon, bound to a carrier molecule called coenzyme A, becoming acetyl-CoA. The key that opens the next phase of energy release, inside the mitochondrial matrix, the innermost space. A cycle begins to turn. It is called the Krebs cycle, or the citric acid cycle, and it is a dance of carbon, hydrogen, and oxygen, a spinning wheel of reactions that release electrons from their bonds, sending them forward to feed a deeper fire. These electrons are not sparks in the way we see light, but they are full of potential, and the mitochondrion knows how to use them. One by one, they are passed into the electron transport chain. A series of protein complexes embedded in the inner membrane, each one more electronegative than the last. The electrons fall through this chain, like water down a series of steps, and with each step, a little energy is captured, used to pump protons across the membrane. From the matrix to the intermembrane space, this creates a gradient, a difference in concentration, in charge, a kind of tension, a potential. It is not yet ATP, not yet power, but it is ready. The mitochondrion has struck its spark, and now it waits for the flame to rise. Inside the mitochondrion, a silent force is gathering. With every electron passed along the chain, protons are pumped outward, crowding into the narrow space between the two membranes. This is the birth of a gradient, a dam of potential energy, waiting to be released. And then, like a flood channeled through a wheel, the protons rush back in, but not freely. They pass through a single spinning gateway. A protein complex called ATP synthase. It is a molecular turbine nestled in the mitochondrial membrane, turning the invisible force of proton flow into the creation of ATP, the universal energy currency of the cell. Each turn of the enzyme's rotor binds together one molecule of ADP and a phosphate, forming a new ATP, like sparks caught and shaped into flame. This is oxidative phosphorylation, the final, most powerful step of cellular respiration. Here, most of the cell's ATP is made, not in the cytoplasm, but here, deep in the folds of the mitochondrion, where breath becomes motion. And it is breath, because at the very end of the electron transport chain, there waits a final acceptor, a molecule that catches the spent electrons, takes on the protons, and becomes water. That molecule is oxygen. This is why we breathe, and not to cool the blood, not merely to move air, but to feed the mitochondria, to receive the electrons and let the fire burn clean. Each breath you take fuels this ancient engine, one that runs not on gasoline or coal, but on glucose and air, bound together in a chemistry as old as life itself. It is silent, it is continuous, and it keeps you alive, every neuron firing in your mind and every muscle fibre stretching beneath your skin, every pulse of your heart, every blink of your eye, runs on this quiet fire, and that fire burns here, in the deep folds of the mitochondrion, unseen, unheard, but never resting. This is not fire in the way of wood and smoke, but fire in its purest form. A controlled release of energy, drawn from the bonds of carbon, guided through the careful channels of life. Each molecule of glucose, once part of a fruit or a grain or a leaf, is dismantled with elegance. Its electrons harvested, its carbon atoms exhaled as carbon dioxide, its energy transformed into ATP. And those ATP molecules, each one a tiny burst of potential, travel through the cell like lanterns, lighting the way for every function. They open ion channels, they power molecular motors, they drive the assembly of proteins, the copying of DNA, the contraction of muscle, the signaling of neurons. With them we move, we think, we remember, we dream. And when they are spent, when that final phosphate is cleaved and the energy released, the cycle begins again. Glucose enters, oxygen flows, electrons fall, protons are pumped, ATP is forged anew. All of it, in rhythm, in silence, in flame. And yet this is more than chemistry, because the mitochondria do not only power life, they help shape its course. They play roles in apoptosis, the quiet self-destruction of cells that are damaged beyond repair. They help manage calcium signaling, vital to everything from heartbeat to memory. They are even involved in the generation of heat, warming the body in times of cold. They are not just power plants, they are regulators, orchestrators, sentinels, and they are ours, but also, in a way, not. For they carry their own DNA, their own memories, their own lineage. Long ago they were separate beings. Free living bacteria drawn into the earliest ancestral cells. And instead of being consumed, they stayed, they offered their energy, and in exchange they were protected. That partnership, forged over 1 billion years ago, still lives in you now. Every mitochondrion in your body carries the whisper of that origin. It speaks in genes passed down only from mother to child, a maternal thread stretching backward in time, generation after generation. Back to the first human. Back to the first cell that ever dared to carry flame. And so we are not powered by machines, but by ancient kin. By once wild fire, now tamed, now woven into the fabric of our being. They're with you always, in your brain and in your lungs and in your fingers and in your breath, tiny flickering embers. Keeping the dark at bay, keeping the rhythm, keeping you alive. You are a being made of flame, but not the kind that burns or consumes or destroys. The kind that builds, that heals, that thinks, that remembers. The mitochondrial flame is the light within. Within the cell, beyond the organelles, beyond the scaffolds and flames, beyond the coded libraries and quiet factories, there is a space, not empty, but full of motion, full of potential, full of life. This is the cytoplasm, the inner ocean of the cell. Not water, but water filled with purpose, a fluid alive with information, a medium dense with meaning. It is not a void, it is a colloid, thick with proteins, crowded with ions, brimming with sugars and salts and suspended chains of function. It is viscous, crowded, electric, a material halfway between fluid and gel, but structured yet in motion, shaped not by walls but by the flow of molecules and the press of constant interaction. Here the rules are different. Space is compressed, time is compressed, chance is amplified. The cytoplasm does not hold still. It cannot. It pulses with molecular waves, vibrations stirred by heat, driven by diffusion, guided by gradients. There are currents and eddies, too small for any eye to see, yet just as real. As rivers on a map, organelles drift, but not aimlessly. They move along paths, marked by probability and intention, directed by scaffolds of protein, guided by motor molecules, with destinations encoded in their structure. Molecules find one another, by chance and by design, a stray ion, a floating enzyme, a cofactor with one purpose only, to meet its partner, to make something new, to start a cascade. Everything is in motion. Everything is in flux. The cytoplasm is not a container, it is the context, the medium of becoming, the stage where life's processes unfold, like actors moving across a floor, that shifts beneath their feet. To stand within the cytoplasm is to stand in a crowd. Every cubic micrometer holds millions of molecules, each with a purpose, each with a destination, even if it doesn't know it yet. Imagine walking through a festival, but every passerby is a protein. Every brush of contact is a signal. Every touch, a chemical possibility. This is macromolecular crowding, a concept as dense as the cytoplasm it describes. Here space is not open, it is saturated. Molecules do not wander freely. they navigate through a thicket of forms and forces. In such a crowded field, reactions that might take hours, in a glass of water, happen in seconds. Because proximity makes everything more likely, because in this place, you cannot help but collide. But it is not chaos. There are pathways, zones of higher concentration, regions where certain tasks repeat like rituals. Some molecules are free agents, others tether themselves to structures, to membranes, to enzymes like anchors dropped into the tide. There are regions of order where factories assemble proteins, and zones of interpretation where signals are decoded. This is dynamic order, not rigid, not planned, but always adaptive, a kind of bustling peace, held together by interaction, by probability, by time, threaded through this ocean. Our filaments, the cytoskeleton, not bones, but cables of protein, actin, microtubules, intermediate filaments, each one with a structure, each one with a role, each one alive. Actin forms thin, flexible networks beneath the membrane, a lattice that allows the cell to crawl, to stretch, to embrace. It is the structure of reaching, the foundation of motion, the whisper of tension just beneath the surface. Microtubules are straighter, stronger, hollow tubes that span the cell's interior, acting as railways, as guideposts, as scaffolds for change. Motor proteins walk along them, carrying vesicles, towing mitochondria, positioning chromosomes, and intermediate filaments, tough tensile ropes, resist pressure and strain. They hold the nucleus in place. They stabilize the shape of the cell, anchoring the inner world, like moorings in a storm. And none of these filaments are fixed, even as you sleep. Even now, they grow, they shrink, they branch and rebuild. It is a cytoskeletal ballet, a performance without curtain or applause, a choreography that began before you were born, and will continue until your last breath. What does it mean for a cell to be alive? It means flow, not just of fluid, but of information, of signals passed hand to hand, molecule to molecule, across a sea that never rests. Within the cytoplasm, messages leap and scatter, like sparks across synapses, calcium pulses, brief and bright, like intracellular lightning. It travels in waves, telling membranes to open, enzymes to activate, muscles to contract. Other messengers slip through the crowd, lipid signals, second messengers, phosphorylation cascades, each one reshaping what comes next. Even mechanical stress, a tug on the cytoskeleton, a pressure at the membrane, can become a message, transduced into biochemical change. In this way, the cytoplasm feels, it responds, it remembers. It is the bloodstream of the cell's inner world, though it does not flow in veins. It is the atmosphere of the cellular city, dense, warm, kinetic, thick with presence, rich with reaction. It is alive, though it has no form of its own. It is not a place the cell occupies. It is the place the cell is. And it is here, in this fluid, invisible tide, that the great dramas of biology unfold: replication, signal, death, survival, joy even, in its most molecular form. To understand the cytoplasm is to understand the background hum of life, the part that never stops, never sleeps, and never asks to be seen. It simply moves. And in doing so, it keeps you alive. Beneath the surface of the cell, beneath the drifting vesicles and pulsing organelles, there lies a hidden structure. Not solid, not static, but living. A dynamic web of filaments and cables, scaffolds and rails, stretching across the cytoplasm in constant flux. This is the cytoskeleton. The cell's secret framework, at once support system and superhighway, architecture and engine. Unlike the rigid bones of the human body, the cytoskeleton is in motion. It bends and reforms. It extends. It withdraws. It is made not of calcium, but of protein. Long thread-like polymers that assemble and disassemble on demand. There are three primary types: microfilaments, intermediate filaments, and microtubules. Each plays a different role, and each contributes to the complex choreography of cellular life. The The microfilaments are the finest threads, woven from actin, a protein capable of both strength and subtlety. These filaments line the cell's edges, helping it hold its shape, but also enabling it to move. When a white blood cell glides toward an invader, or when a fibroblast crawls across a wound, it is the actin filaments that push the membrane forward, ripple by ripple. In a process called cell crawling, intermediate filaments, thicker and more stable, serve as the internal girders of the cell. They resist tension, they keep the nucleus in place, they stretch across the cytoplasm like suspension cables, anchoring organelles, and giving the cell resilience against mechanical stress. Without them, the cell would deform with every twitch and tug from the world around it. And then there are the microtubules, hollow tubes, broad and strong, built from units of a protein called tubulin. These are the true highways of the cell. They radiate out from a central organizing center near the nucleus, forming tracks upon which cargo is moved. Organelles glide along them. Vesicles travel their length. Even chromosomes, during the great dance of cell division, are guided by microtubules. But the cytoskeleton is more than a scaffold. It is a system of communication, a means for the cell to sense its shape, to measure its interior, to respond to forces and signals. It is responsive, intelligent in its own quiet way, assembling in one moment, vanishing in the next, always in dialogue with the needs of the cell. And all of this, the pushing, the pulling, the transport, the structure, happens in silence, without wires, without wheels, only threads, flowing and reforming in the fluid dark. The cytoskeleton is the reason the cell has a shape. It is the reason it can move. It is the reason it knows where it is. Without it, the cell would be a droplet, amorphous and lost. But with it, the cell becomes a city, defined, responsive, and alive. Within the lattice of the cytoskeleton, something remarkable unfolds. Motion, deliberate and directed. The cell is not a passive place. It is a city, alive with traffic. And along its internal highways, cargo is constantly on the move. These are not random drifts or thermal jitters. This is active transport, and it is driven by microscopic machines that walk. The couriers of the cell are proteins, specialized motor proteins, and they do not float, they stride. Some, like kinesin, move toward the outer edges of the cell. Others, like dynein, head back towards the center. Each one walks along the microtubules, stepping hand over hand, 1 foot of protein binding to the tubulin track, then lifting and swinging the next. With each step, they consume ATP, the same cellular energy made in the mitochondria, powering their march a few nanometers at a time. Attached to these walking proteins are vesicles, bubbles of membrane that hold everything the cell might need to deliver. Some carry newly formed proteins, bound for the surface or another organelle. Others transport lipids, enzymes, or molecular signals, meant to coordinate the activities of distant regions within the cytoplasm. Even mitochondria themselves, the powerhouses of the cell, can hitch rides along these tracks, moving where energy is most needed. And when it's time for the cell to divide, the motor proteins shift their focus, helping to organize chromosomes, stretch the spindle, and pull the genetic material into 2 symmetrical halves. But all of this occurs in silence. There is no sound here, no clatter of hooves, no rattle of wheels, only the careful footfalls of proteins. Each step planned, each destination known. And this transport is not chaotic. It is regulated by signals, chemical tags and protein scaffolds that guide cargo to the right locations. Vesicles destined for the membrane are pulled outward. Others are rerouted to the Golgi or returned to the endoplasmic reticulum. The motor proteins recognize these destinations, not by sight or map, but by molecular signs, like postal codes inscribed on their surface. In this way, the cell becomes something extraordinary, not merely a sack of chemicals, but a place of logistics, a place where delivery, timing, and accuracy matter. There is a poetry in this motion, a choreography performed beneath all thought, and though you never feel it, though you cannot hear it, this inner traffic is what allows you to feel, to move, to be. Every heartbeat, every memory is built upon this steady procession. Cargo walking along roads no wider than a whisper. The cytoskeleton is not a fixed scaffold. It is not a framework set once and left to endure. Instead, it is alive, growing, shrinking, shifting in response to the needs of the cell. It is an architecture in motion, a fluid structure that adapts in real time, responding to both internal decisions and external cues. Consider how a cell changes shape, not just in growth, but in migration. A white blood cell chasing down a bacterium, a neuron stretching its axon toward a distant signal, a fibroblast crawling into a wound to help heal the skin. None of these motions are possible without the cytoskeleton reshaping the cell from within. The actin filaments at the cell's edge push forward, assembling rapidly to extend the membrane. Behind them, myosin motor proteins pull against the actin like microscopic muscles, contracting the rear of the cell, pulling the body forward. Together, this creates a kind of cellular locomotion, slow and silent, but incredibly purposeful. Even the microtubules are not static. They grow and shrink dynamically in a process called dynamic instability. At any moment, a microtubule may suddenly extend, probing a new area of the cell, or it may collapse, retracting as the cell adjusts its internal map. This constant remodeling allows the cell to change direction, reorient its internal pathways, and respond to stress or opportunity. When the cell divides, this dynamic architecture becomes even more dramatic. The microtubules reorganize into a structure called the mitotic spindle, which attaches to the chromosomes and carefully pulls them apart. It is one of the most precise operations in biology, separating two identical sets of DNA without tearing or misplacing a single thread. All of this, the crawling, the pulling, the dividing, relies on the same proteins, assembling and disassembling over and over, in countless combinations. It is as if the cell is built each moment anew, from pieces that never fully come to rest. And while it may seem chaotic, there is a deep intelligence in this, not of thought, but of balance. Feedback loops and signaling cascades guide the construction. Protein concentrations determine the rate of growth. Mechanical tension influences direction. The architecture is not planned in advance, it is felt, responded to, grown from within. In this way, the cytoskeleton becomes something more than a scaffold. It is the cell's awareness of its own form. It is the reason a cell knows where it ends, and the world begins. Beneath the shifting scaffolds and the restless filaments, something deeper begins to emerge. The cytoskeleton is not just a structure. It is a history, a memory written in tension and form, a record not kept in ink or code, but in shape and direction. Cells remember where they've been, not through conscious thought, but through the trails laid down by their filaments. When a neuron sends out its long axon, seeking connection, it is guided by cues in its environment, but it is also guided by its own past. The growth cone at its tip feels, responds, chooses, but behind it, the cytoskeleton hardens into a path, marking where it has already reached. Even single-celled organisms remember. A paramecium, though it has no brain, can change its behavior in response to touch. The cytoskeletal elements beneath its membrane reorganize, altering the cell's motion, building a temporary memory of experience, a way of saying, I have been here before. In multicellular organisms, this becomes more complex. Cells must orient themselves to one another. Skin cells align to form a barrier. Muscle cells contract in unison. Epithelial cells curve into the shapes of glands and lungs. All of this requires internal guidance. All of it depends on the cytoskeleton, knowing not only how to form, but how to align, how to join, how to contribute to something larger than itself. And sometimes that memory lingers. In stem cells, the cytoskeleton helps guide differentiation. shaping the future identity of the cell. Even after division, traces of polarity, of alignment, of stress can influence how a daughter cell behaves. The structure you inherit shapes the choices you make. This may be why the cytoskeleton is implicated in so many human diseases. When it falters, so does the cell's sense of direction. Cancers lose their shape, begin to grow without form, neurons lose their structure, and signals fade. The breakdown of these threads is more than mechanical. It is a kind of forgetting. And yet, in every healthy cell, the cytoskeleton continues its quiet labour, holding shape, bearing memory, marking trails, offering the cell not just support, but a sense of self. You are built, not only of molecules and genes, but of motion, of shape, of memory, in filamentous form. The highways within you do not simply carry things. They carry knowledge of where you are and where you've been, threads that remember and keep you whole. At the edge of the cell lies its most ancient boundary, a delicate shimmering veil that separates self from other, life from world. This is the plasma membrane, the first gatekeeper of life, the sentinel of every cell. It is not solid, yet it holds everything in. It is not static, yet it maintains form. Composed of a bilayer of phospholipids, its surface undulates softly, like the surface of a living lake. Flexible, fluid, endlessly in motion. Each phospholipid molecule is shaped like a matchstick, a round, water-loving head, and two slender, water-fearing tails. In water, they align into two sheets, heads facing outward toward the watery world, tails tucked safely inward, away from both the outside and the cell's interior. This structure creates a paradox, a boundary that is both strong and supple, firm enough to preserve identity, yet flexible enough to bend, reshape, and respond. Floating within this liquid fabric are proteins of many kinds, some embedded fully from one side to the other, others resting just on the surface. Some of these proteins act as bridges, linking the inside of the cell to the outside world. Others drift like boats on a tide, carrying messages, ferrying cargo, or anchoring structures in place. This ever-shifting combination of lipids and proteins is known as the fluid mosaic model, a living, breathing border capable of sensing, responding, and adapting. The membrane is selective. It does not permit the world to rush in unchecked. It speaks in the silent language of channels and pumps, of electrical gradients, and molecular recognition. Sodium and potassium ions, vital for electrical signaling, move only through tightly controlled channels, tiny passageways that open and close in response to voltage, mechanical force, or chemical signals. Each ion, each molecule must pass a kind of molecular test. Its size, shape, and charge scrutinized before entry or exit. Calcium, when allowed to enter, acts as a spark, triggering cascades of internal events, like ripples from a single drop of water, falling into a still pond. Glucose, the sugar fuel of life, must be escorted. It knocks at a special door, a transporter protein, that recognizes its subtle geometry and ushers it inside. And even water, so simple, so abundant, cannot pass freely. It flows through gated channels called aquaporins, tubes so selective. They allow only the right flow at the right time, like valves in a submarine hull. This is not a wall. It is a sensor, a negotiator, a diplomat. The cell is not a prisoner behind its barrier. It is a participant in the world, opening and closing its borders with exquisite control. On the outer surface of the membrane, receptor proteins rise like antennae, sensitive tendrils reaching into the unknown, waiting, listening. A hormone may arrive, a messenger molecule shaped by a distant gland, and bind to the receptor with a precision like key meeting lock. A neurotransmitter may touch its counterpart briefly and perfectly. And suddenly a change begins inside. The receptor does not enter the cell. It does not carry anything across. Instead, it translates presence into meaning. Like a watchtower scanning the horizon, it senses danger, or invitation, or instruction. The signal is passed inward, into a network of internal proteins, a relay of molecular switches and messengers that amplify, redirect, and interpret. Some messages are simple: grow, move, divide, rest. Others are nuanced: too much light, not enough glucose. Begin repair. Stay calm, all is well. Through these communications, cells coordinate with one another, forming tissues, organs, and entire bodies that pulse and breathe in unison. But the cell does not only listen, it also speaks. And when it needs to send a message outward, it does so in vesicles. Small spheres of membrane, budding from within like soap bubbles, carrying chemical notes, wrapped in protective skins. These vesicles travel to the cell's outer edge, guided by motor proteins and the cytoskeletal tracts. When they reach the membrane, they fuse, becoming one with the border, and release their contents into the space beyond. This is how neurons communicate, releasing neurotransmitters across the tiny gap between one cell and the next. It's how hormones enter the bloodstream, delivering instructions to distant cells and tissues. It's how immune cells call for help, summoning reinforcements to sites of injury or invasion. Even the act of engulfing, of taking in a foreign body, a nutrient, or a dying neighbour, is done through this membrane. It folds inward, embraces the visitor, and draws it into an internal vesicle for study, digestion, or protection. In all of these roles, the plasma membrane becomes something astonishing. Not a wall, but a platform for exchange. Not a shield, but a translator of signals. Not merely a boundary, but a bridge between life and its surroundings. The gatekeepers of the cell are not just guards, they're engineers, interpreters, artisans of interaction. And through them, the boundary between self and world becomes not a barrier, but a doorway, alive with meaning, sensitive to change, and essential to everything the cell does to survive. There comes a time deep within the rhythm of every living cell when growth alone is not enough, when it must become two. It does not rush this moment. It waits, it listens, it measures its state and the space around it, as if asking the body, the world, "Am I ready?" Then quietly, without a sound, the process begins. This is mitosis, the division of the self. It is not destruction, it is reproduction, a passing on of memory, of architecture, of possibility. A single cell, fully alive and whole, must become two, equally whole, equally alive. And in doing so, it creates continuity. Not the continuity of a thing, but of a process, of life itself. The cell does not vanish in division, it transforms, it unfolds, it becomes something more, by becoming again. The first step is preparation. Inside the nucleus, the DNA, that long coiled thread of instruction, begins to duplicate. Every gene, every chromosome, each one carefully copied, by enzymes that read, match and double-check the code, with breathtaking precision. These two identical copies, called sister chromatids, remain attached at their centres, like twin scrolls bound together, waiting for the moment of parting. Outside the nucleus, the cytoskeleton begins to shift. The microtubules, those dynamic filaments that once shaped the cell and carried cargo, reorganize with purpose. They begin to form a spindle apparatus, a temporary scaffold of threads, whose task is singular and sacred, to move chromosomes. But the cell does not leap into action. It pauses. It checks everything. Has the DNA been copied correctly? Are the chromosomes intact? Is there enough energy, enough building blocks, to create not one but two? Only when every signal is clear, every system confirmed, does the cell move forward. This is the elegance of control. This is the choreography of life. Nothing is left to chance. As mitosis truly begins, the nuclear envelope dissolves, not with violence, but with calm disassembly. What was once hidden now becomes visible. The chromosomes, so long loose and interwoven, condense into tightly coiled forms, distinct and structured, ready for travel. They align at the center of the cell, along an invisible plane called the metaphase plate. a perfect line drawn across the equator of life. Each chromosome, still paired with its twin, is attached at the centromere to fibres of the spindle, as though holding a taut ribbon, waiting for the cue to let go. And then, with a signal as silent as breath, the separation begins. The spindle fibres begin to shorten, pulling each sister chromatid toward opposite poles. Not with chaos, but with certainty, with a momentum as inevitable as gravity. It is a dance, slow, deliberate, powerful, but one without audience, without applause, without witness. A performance carried out 1,000,000,000,000 times a day, every day, inside you. Each time flawlessly rehearsed, yet utterly unique. Finally, when the chromosomes have reached their destinations, the cell begins to rebuild. 2 new nuclear envelopes form, gentle casings, around each newly organized set of DNA. The chromosomes, no longer needed in their coiled state, begin to unravel, returning to their soft open form, ready once again to be read and to be active and to be alive. Meanwhile, the cytoplasm itself divides. This step, called cytokinesis, is like the tying of a ribbon around the cell's middle. A band of actin and myosin tightens, drawing inward like a belt, until the single form cleaves cleanly in 2. And there, where once stood 1, now stand 2. 2 daughter cells, identical in their essence, yet free to grow in their own direction, to take what was given, and build from it something new. This process, so fluid, so ancient, is how you were once formed. From a single fertilized egg, your body unfolded, grew, and blossomed, through billions upon billions of such divisions. And even now, in your skin, in your blood, your bones, the cycle continues. Cells grow, divide, replenish, repair. Mitosis is the quiet miracle of becoming, the act by which life sustains itself, not with noise but with rhythm, not with drama but with grace. It is the cell's way of saying, life must go on, and it will, in silence, in symmetry, in endlessly unfolding beginnings. You are not made of stone or flame, not of wire or glass, but of cells, trillions of them. Each one a living unit, a tiny engine of purpose. Skin cells that flake and renew, muscle cells that pull and lift, nerve cells that stretch across impossible lengths, transmitting thought in waves of voltage, blood cells that tumble through arteries and veins, delivering oxygen like silent couriers. Every cell is a complete world, with its own walls, its own rules, its own secrets. And yet, they do not act alone. They coordinate, they communicate, they harmonize. Together, they form tissues. Tissues build organs. Organs create systems. And those systems become you. But it all begins with the microscopic, with the folding of proteins, with the motion of a motor protein walking a vesicle across a filament, with a strand of DNA being copied, read, and passed on. You are the sum of these parts, not only in body, but in history. The cells that make you today carry instructions first written hundreds of millions of years ago, in ancestors who crawled from oceans, who breathed air for the first time, who saw with eyes formed from light-sensitive cells. There is no part of you untouched by this cellular memory. The way your fingers heal, the way your stomach digests, the way your eyes adjust to moonlight. You are not simply living. You are being continually remade, and the makers are the cells. At every moment, without pause, your cells are working. Even as you sleep, even as you drift into dreams, they carry on, unseen and unsung. In your heart, cardiac muscle cells contract in perfect synchrony, triggered by electrical waves that ripple across the tissue. In your lungs, epithelial cells form the barrier between breath and blood, exchanging oxygen and carbon dioxide with every exhale. In your liver, enzymes process nutrients, clear toxins, and manage your store of energy like an accountant balancing a ledger beneath your awareness. Every second, somewhere in your body, a protein is folded. Somewhere else, a ribosome begins translation. A mitochondrion spins its ATP-producing turbine, and a membrane gate opens to admit a burst of sodium. There is no conductor to this orchestra, and yet the symphony plays on. Cells sense their neighbours. They respond to hormones in the blood, to neurotransmitters drifting through synapses, to mechanical tension, chemical gradients, and subtle electrical cues. They're not merely alive, they're aware in their own molecular way. Not with thoughts, but with feedback loops and signal cascades. Not with memory as you know it, but with gene expression patterns. With epigenetic marks, with networks of cause and effect, that ripple outward through tissues like rings in a pond. This, then, is your life from the inside. Not one great machine, but billions of tiny ones. All humming together, each with its own rhythm, its own purpose, its own grace. You do not command them. They are not waiting for your instruction. They are you, at a deeper level than thought, deeper than language. They are the pulse beneath your pulse. The body you wear is not the one you were born with. It has changed, quietly, cell by cell. Skin renews itself every few weeks. Blood cells are replaced every few months. The lining of your stomach, exposed daily to acid and enzymes, turns over even faster. And yet, you remain. Your shape, your voice, your way of moving through the world, all persist, carried not by the permanence of matter, but by the continuity of form. This is the miracle of cellular renewal, that what is constantly dying is also constantly being made. A kind of immortality, not of any one part, but of the pattern they uphold. When a cell grows old, when its DNA is damaged beyond repair, when it fails to carry out its duty, it does not cling to existence, it lets go. There is a name for this: apoptosis, programmed cell death, an act not of violence but of order. The cell packages itself, signals for cleanup, and dissolves in peace. It is not failure, it is wisdom. A way for the body to maintain its clarity, to make space for what comes next. Other cells take its place. Stem cells divide and specialize. Tissues mend, bones knit. The memory of the structure remains, even as its parts are remade again and again. In this you're like a flame, never the same molecules, but always the same light. There is something quietly astonishing in knowing what you're made of, that beneath your thoughts, your choices, your dreams, there is a hidden world, humming, folding, copying, moving. That every breath you take is supported by molecules that walk, membranes that shift, signals that flash across cellular landscapes like constellations coming to life. You are not built from magic, and yet it feels like magic, doesn't it? You are not a single thing, but a society of selves. 1,000,000,000,000 cities working in harmony, each cell its own kingdom, each organ its own culture, each moment held together by the quiet labour of the invisible. And through all of this, through the mitochondria spinning energy, through the ribosomes stitching protein, through the membranes exchanging signals with the world, you live. You feel sorrow, joy, hunger, wonder. You speak, you sleep, you imagine the stars, all while these tiny machines labour faithfully within you, unaware of the body they support, yet essential to its every moment. This is the dream of being alive, to be made of matter, and yet to wonder what it means, to carry oceans within a single drop, to be old as time, and yet brand new with each breath. You are not separate from your cells. You are not merely their passenger. You are their song. And as you drift now towards sleep, let them carry you gently, like the tides inside a shell, like the rhythm of a dream. You are made of this, and it is beautiful. Another journey comes to an end. Our time within the cell, among threads and tides of life unseen, has brought us closer to the quiet brilliance that lives inside us all. We've wandered the scaffold of the cytoskeleton, listened to the breath fire of the mitochondria, watched the ribosomes stitch the alphabet of life, into form, into function, into memory. These are the silent miracles, happening within you right now. Softly, faithfully, whether you are awake or already dreaming. Here at Midnight Voyager, we believe that wonder never truly sleeps. It only changes shape, drifting from thought to dream, from question to calm. If you've made it this far, perhaps your eyelids are already heavy. Perhaps the world has grown quiet around you. If you're still awake, I invite you to leave a gentle note in the comments. What image stayed with you? What moment lingered? And if you're drifting, don't worry, there are more journeys waiting. Let the playlist carry you from one voyage to the next. Each story crafted to calm the mind, soothe the heart, and invite sleep in like a trusted friend. Until next time, rest well, dream deeply, and thank you for joining us on this midnight voyage.